to what diffusion is. So a solution, this consists of a solvent. Typically within the body, we consider water to be our solvent. And then also for some type of solute, which is going to be some type of molecule that will dissolve in water. So molecules in a solution then are going to be in a constant state of motion. They are not static, but they are rather dynamic. And so if there is a concentration difference between two regions, so typically we think of inside the cell versus outside of the cell, random motion will establish equilibrium via diffusion. And so diffusion will occur without a physical separation or across a permeable membrane. And so with diffusion, this concept obeys the second law of thermodynamics because diffusion actually increases entropy. And so this process is crucial to the survival of every cell, and it plays an important role for exchange of, say, oxygen and carbon dioxide between blood and air and lungs, and then also for movement of substances across the kidney tubules so that we are able to hold on to what we need and then get rid of what we do not need. So with diffusion, we are going from an area of high concentration to an area of low concentration. That's going to be the predominant movement that we see in terms of molecules. However, there will be some movement back and forth where we'll go from the lower concentration into that of the higher concentration, just not nearly as great as that from the high to the low. So what we have is net diffusion then, where this is going to be due to random movement, and the net direction of diffusion is going to be from high to low. Okay, and so as you can see here in panel A, we have a higher concentration of solute, and that is going to move to this area of lower concentration of solute. And so this will continue on until there is no net diffusion occurring, where we'll have equal concentrations on both sides for the left and the right. However, there will still be movement between the molecules. It doesn't mean that they stop once that they've migrated over. So when we look at the body, we have diffusion occurring through the plasma membrane. And so what we have are small and nonpolar. If you remember, nonpolar are going to be uncharged, okay? So meaning that they don't have a plus or minus associated with them. And also lipid soluble because of that lipid bilayer. Molecules that are going to pass easily through the lipid portion of the membrane. And so examples of this include oxygen and carbon dioxide, and also small steroid hormones. And so this is important because if you think about gas exchange, we need net diffusion of oxygen into the cell, so like what we have represented in this picture, and then we need rapid diffusion of carbon dioxide outside of the cell. And so this is going to be established, again, by differences in concentration gradients where they go from high concentration to low concentration. Water, however, is a polar molecule, so it passes through special channels called aquaporins through osmosis. So when we look at this picture, what we have are for molecules that are either going to be charged, large, and that are polar, which again would have a charge associated with them, or hydrophilic, these require then different types of channels to enter by diffusion from extracellular environments into an intracellular environment. Okay, and so again, the concept though of diffusion is that it's going from high concentration to low concentration. However, the only difference now is that we need to have some type of channel that allows that to occur for these types of molecules and ions. There are several variables that will affect the rate of diffusion. And so these are going to be the four that will affect it. So an increased magnitude of a concentration difference will increase the rate of diffusion. So if we have, say, a very high concentration on one side and a very low concentration on another, that greater magnitude will then favor in rapid rate of diffusion. Also, we'll have an increase in permeability of the membrane. So if we insert more channels or aquaporins, that will also increase then the rate at which diffusion occurs. A warmer temperature will also increase the rate of diffusion, whereas a colder temperature will slow down the rate, and that has to do with kinetic energy. And then also for cells, a larger surface area of that membrane will also allow for more diffusion to occur because of an increase in permeability, number of channels that are found, but also the just vast amount of molecules that can penetrate by osmosis or diffusion. But the driving force, though, that is going to affect the rate of diffusion is going to be this first one, so the magnitude. So what is the concentration difference will really affect then the rate of diffusion that occurs. So with osmosis, this is a little bit different, where now this is diffusion of a solvent instead of a solute, okay? And so these are going to be aided by channels in the membrane called aquaporins, and typically, again, our solvent is going to be water. And so water molecules do not carry a charge, so they can pass through the plasma membrane slowly. There has to be, though, a solute concentration difference on either side of a membrane that is has to be permeable to water. And so the membrane must be impermeable to the solute, or the concentration difference will not be maintained. So we only will have the movement then of the solvent or of water. Solutes that cannot cross and permit osmosis are going to be considered osmotically active. And so many aquaporins we find within the kidneys, the eyes, the lungs, salivary glands, and then also within the brain, so that we have that net movement of water. So when we look at this image here, what we have is the water molecules are represented by these little blue dots, and then these solutes are going to be different types of molecules. So in terms of osmosis, we have a higher concentration of our solvent of water on the left side. This yellow here represents a membrane, and then so water is going to move then from this left chamber into that right chamber. Now, again, it has to be permeable within the membrane to our solvent of water. However, there has to be an impermeability to the solute. So meaning the solutes are not able to move then from their concentration gradient of lower to higher, okay? So that's why in this figure, even though I'm drawing all over it, let's erase all that, what you don't see are large holes, right, for the solute. So that means that we are impermeable to our solute. With osmosis, this leads us to the concept of tonicity that occurs with cells. And so this will determine whether cells remain the same size, will swell or shrink when a solution surrounds the cell. So when we have an isotonic solution, iso means the same. And so it has the same concentration of non-penetrating, so no movement of solutes as normal body cells. And so this means that the cell volume will remain constant. Again, because the solutes are not able to move, it's just the solvent. A hypotonic solution will have lower concentrations of non-penetrating solutes than normal body cells. And so this will cause then for water to enter the cell, causing it to swell or sometimes to rupture. So hypotonic refers to the concentration of solutes, a low solute concentration. Okay? Hypertonic solution means that there are a higher concentration of non-penetrating solutes than normal, and so we'll have less water, and so the cells are going to shrink as they lose water by osmosis. So here, when we look at the difference, this is a representative of a red blood cell, and this is a great example of where this would come in useful. So in an isotonic solution, we have the same volume, okay? So the same amount of water entering and exiting the cell is going to be similar so that there's no net change. In a hypotonic, again, hypo means that we have a lower concentration of solutes than normal. So what we'll have is the entering of water into that cell, causing that cell to swell. This can become a problem because that can rupture the cell, and if the cell ruptures, that means that it is not irreversible. Hypertonic solutions, though, have a higher concentration of solutes, and so water is going to rush out of that cell into that hypertonic solution, causing that cell to then shrink or crenate in size. Okay, and the good thing though is that this one is reversible, so that if you add water back and put it into a hypotonic solution, then what will happen is that the cell will increase in size once more.